In Unit 3, Lecture 2, Part 1, we're going to be looking at the role the ability of a bacterium has to compete for iron and nutrients, and the role that plays in its ability to colonize the body. So once again, don't forget to read your bullet points at the beginning of each of these SoftChalk lessons and learn your detailed learning objectives. So remember our topic we're currently talking about, we're looking at virulence factors that promote bacterial colonization of the host. And we've already looked at several of these. We're currently down to number 4 now, the ability to compete for iron and other nutrients. So as we learned earlier in the semester, for a bacterium to colonize an area in or on the body, that location of the body must have the correct physical and chemical environment. That is, an environment that has the proper amount of oxygen content, the right pH, the right nutrients, the right temperature, and such. And those are the things that would be needed for that pathogen to multiply in that part of the body. So often the ability to be pathogenic is directly related to the bacterium's ability to compete successfully with host tissue and with the body's microbiota for limited nutrients. So keep in mind all the microbiota that live in and on the body, and they wouldn't be microbiota if they couldn't compete successfully with our cells for the same food. When a pathogen enters the body, it has to not only compete with the microbiota, but with our cells for the same nutrients. And that's one of the reasons why the generation time, remember that's the time it takes for a population of bacteria to double in number, the reason for the generation time of bacteria growing in the body is substantially slower than in lab culture, like in a test tube, because in the body, the essential nutrients are limited. Whereas, of course, in a test tube initially, there would be plenty of nutrients available. And of course, we also have to consider in the body that body's defenses are also removing microorganisms. So one of the major reasons why the overwhelming majority of bacteria found in nature are not harmful to humans is simply they can't grow well in or on our body. They can't compete. That isn't a good environment for them to live. So as we've said, to be pathogenic, a bacterium has to be able to multiply in host tissue. And the more rapid the rate of replication, the more likely the infection will be established. So pathogens have to be able to compete successfully for the limited nutrients in the body. Again, compete with human cells for those nutrients and with microbiota for those nutrients. Now, bacteria generally compete for nutrients by synthesizing specific transport systems or cell wall components that bind limiting substrates like a particular nutrient and transport them into the bacterial cell. And a good example of that we're going to use in this SoftChalk lesson is the ability of bacteria to compete for iron. Now, as part of our innate immunity, as we'll learn in Unit 5, the body goes to a lot of trouble to deprive microorganisms of iron. And that's because iron is an essential for both bacterial growth and human cell growth. Iron is a very common cofactor in enzyme reactions, and both bacteria and humans need that iron. Iron is also, remember, a key part of the electron transport system. So bacteria synthesize what are called iron chelators. And a chelator, iron chelator is a compound that's able to bind iron. And in bacteria, those iron chelators or iron binding compounds are called siderophores. Many of the siderophores are excreted by bacteria into the environment where they bind free iron. And then once the iron's bound to the siderophore, it re-enters the cell and the iron's released for the bacterial use. Other siderophores are found on the cell wall where they bind free iron and then transport it into the bacterium. So the bacterium does have to compete with human cells and microbiota for the same limited iron that's available in the environment. Well, meanwhile, the body's producing its own iron chelators to trap iron for use by human cells. And we have a number of iron chelators that we produce, including transferrin, lactoferrin, ferritin, and hemin. So the ability of the bacterial iron chelators to compete successfully with the body's iron chelators, as well as those as microbiota or normal flora, are essential to pathogenicity. Now, in addition to making siderophores, some bacteria use some other tricks to get their fair share of iron. Some bacteria produce receptors for siderophores of other bacteria, and in this way they steal iron from other bacteria. Also, some bacteria are able to bind our iron chelators, like transferrin, lactoferrin, ferritin, and hemin, and steal iron basically from us. Our iron chelators bind the iron, but then the bacteria bind our iron chelators and make use of that iron. For example, Neisseria gonorrhoeae that causes gonorrhea, Neisseria meningitidis that causes meningococcal meningitis, and Haemophilus influenzae that causes a variety of respiratory infections are able to use iron bound to human transferrin and lactoferrin for their own iron needs, and those are human iron chelators. Some bacteria produce proteases that degrade human iron chelators, which are proteins. So they can use proteases to degrade lactoferrin, transferrin, and heme. And when these protein chelators are broken down, the iron that they had bound is released, and that can then be captured by bacterial siderophores. And a few bacteria don't even use iron as a cofactor. Borrelia burgdorferi that causes Lyme disease uses manganese as a cofactor rather than iron. And then finally, some bacteria are able to produce exotoxins that kill host cells, but they make these toxins to kill the host cells only when iron concentrations are low. And that way they gain access to the iron that was stored in that cells. And once they have enough iron to start growing again, they stop producing the toxin, they stop killing our cells. So there's a variety of techniques, as we see, that bacteria can use to get their fair share of iron. And keep in mind, for virtually any nutrient that bacterium needs, its transport systems have to compete with our transport systems and that of the local microbiota for the same nutrients. So one of the essential things in the ability to colonize the body, again, is the ability of the pathogen to directly compete with host tissues and microbiota for the same nutrients. And as always, there's a little self-quiz you can do at the end.